A very good morning to everyone who are gathered here. I request all the dignitaries to come over the stage. A very good morning to the respected dignitaries, distinguished alumni, faculty members, respected guests, and my friends who are all gracing this event today, where familiar paths meet once again. In this shared moment, achievements, experiences, and memories come together, shaping a phase filled with moments where time slows down and togetherness takes the lead. It is with great pleasure and humility that I, P. Naveshri, a senior MSc student, exploring the wonders of this academic ecosystem, standing here to take you all through a lane of memorable experience. As we gather today, bridging past and present to honor that legacy, let us immerse ourselves in the anthem that has inspired generations. I kindly request everybody to stand in spirit as we play the university song, a melody that carries the pride, shared journey, and the enduring bonds of this alma mater. A heartfelt thank you for joining in that visual celebration of our university. Now I request all the dignitaries to occupy the seats over the dais. In the vast culture medium of science, some leaves become the primary inoculum, initiating the growth that continues long after the first transfer. To honor such unwavering dedicated stalwarts of our field, second memorial lecture is being organized as a tribute to such enduring legacies. Emphasizing the contributions of Dr. T.K.R. Reddy and Dr. R.B. Patil sir, whose work and vision enriched the discipline of microbiology. Like master cultivators of knowledge, they prepared the fertile ground where curiosity germinated, ideas replicated, and scientific thinking has elevated to its exponential phase. On this solemn occasion, we honor the influence of Dr. T.K.R. Reddy and Dr. R.B. Patil sir within the confines of our department. Before we begin the festivities of this alumni meet, let us embrace a sacred moment of reflection. I am delighted to call Gaganashri, third year PhD student, College of Agriculture, Dharwad, to present the divine melody before all of us.
जय देव श्री गणपतिराय जयकारक भयहारक ओंकारकाय जय देव श्री गणपतिराय जगदंबೆಯ ಮೈಮಣ್ಣಲಿ ಮೂಡಿದ ಮೂರುತಿಯೇ ಮೃಗರ ನೋಡಿದ ಮಾಡಿದ ಬೇರೆಯಾಕೃತಿಯೇ ಜಗದಂಬೆಯ ಮೈಮಣ್ಣಲಿ ಮೂಡಿದ ಮೂರುತಿಯೇ ಮೃಗರ ನೋಡಿದ ಮಾಡಿದ ಬೇರೆಯಾಕೃತಿಯೇ ಚಿಗುರಾಯಿತು ನಿನ್ನಾಲಿಗೆ ದಿನ ದಿನವೂ ಶ್ರುತಿಯೇ ಹಗಲಾಯಿತು ನಿನ್ನೆಲಪೆ ಸುಳ್ಳಾಯಿತು ಧೃತಿಯೇ ಗಣನಾಯಕ ಕರಿನಾಯಕ ಜನತ್ರಾಯಕ ಸಿರಿಯೇ ಗುಣವರ್ಧಕ ಬಲವರ್ಧಕ ಸುರಸ್ಪರ್ಶಕ ದೊರೆಯೇ ಗಣನಾಯಕ ಕರಿನಾಯಕ ಜನತ್ರಾಯಕ ಸಿರಿಯೇ ಗುಣವರ್ಧಕ ಬಲವರ್ಧಕ ಸುರಸ್ಪರ್ಶಕ ದೊರೆಯೇ ಅಣು ಅಣುವು ತೃಣರೇಣುವು ಗುಣಗಣವೆಂದರಿಯೇ ಕ್ಷಣ ಕ್ಷಣವೂ ನಾ ಸಿದ್ಧಳು ನಿನ್ನೊಂದಿಗೆ ಬೆರೆಯೇ ಅಣು ಅಣುವು ತೃಣರೇಣುವು ಗುಣಗಣವೆಂದರಿಯೇ ಕ್ಷಣ ಕ್ಷಣವೂ ನಾ ಸಿದ್ಧಳು ನಿನ್ನೊಂದಿಗೆ ಬೆರೆಯೇ ಜಯ ದೇವ ಶ್ರೀ ಗಣಪತಿರಾಯ ಜಯಕಾರಕ ಭಯಹಾರಕ ಓಂಕಾರಕಾಯ ಜಯ ದೇವ ಶ್ರೀ ಗಣಪತಿರಾಯ ಶ್ರೀ ಗಣಪತಿರಾಯ ಶ್ರೀ ಗಣಪತಿರಾಯ. ಧನ್ಯವಾದಗಳು.
Thank you, Gaganashri, for your soul-stirring rendition of the invocation song. Just as a well-balanced culture begins with the right conditions, every meaningful event begins with warm words that bring people together. To extend that warmth and formally welcome everyone present today, it is my pleasure to invite Dr. V.B. Kuligod sir, Dean Agri and Campus Head, College of Agriculture, Dharwad, to welcome the gathering. Over to you, sir.
Good morning to all. So today, the Department of Agriculture, Microbiology and College of Agriculture and the University together are celebrating the second memorial lecture in the name of late professors T.K.R. Reddy as well as Dr. R.B. Patil. So I am privileged and respectfully I welcome all the dignitaries and the students on the dais. So first of all, we have with us today Dr. D.L.N. Rao, sir, and he has come to deliver the lecture. I welcome warmly as well as respectably D.L.N. Rao, sir, on the behalf of the college, microbiology department, as well as the entire university. So I request our Honorable Vice Chancellor, sir, to welcome him with the floral bouquet. And all our functions are presided over by our Honorable Vice Chancellor. With his advice, with his support, we could able to organize many functions in the Agriculture College, Dharwad. So today, this function is being presided over by our Honorable Vice Chancellor, sir, Dr. P.L. Patil, sir. So I welcome warmly our Honorable Vice Chancellor, and I request Dr. P.V. Krishna, sir, to welcome him with a bouquet. And we have another guest of honor today, Dr. D.J. Bhagyaraj. Joseph Bhagyaraj is here with us, and I welcome warmly with him. And I request our Honorable Vice Chancellor to welcome with a bouquet. And our Honorable former Vice Chancellor, Dr. G.H. Kulkarni, sir, is also here with us. And I request our Honorable Vice Chancellor to welcome him with a bouquet. And our honorable board members are always with us with a lot of support, as well as they always grace our functions. And today with us, Dr. Basavaraj Kundgolmad, sir. And I request Dr. Srinivasulu to welcome him with a floral bouquet. And another honorable board member, Board of Management is here with us, Dr. Maltesh Shagoti, sir. And I welcome warmly Dr. Shagoti, sir. So I request Jones Nirmal Nath to welcome him with a bouquet. And the Director of Research and my colleague, Dr. P.U. Krishna, sir, is also here with us. And I welcome P.U. Krishna, sir, for this function. And I request Dr. Ashwini to welcome him with a bouquet. Thank you, sir. And today's function is being organized by the organizing secretary and head of the department, Dr. C.R. Patil, sir. So I warmly welcome him, and I request Santosh to welcome him with a bouquet. Sir, many of the previous former officers and the students of T.K.R. Reddy and Dr. R.B. Patil, sir, are here with us to grace this occasion. So naming few, Balasubramanian, Dr. S.G. Patil, sir, Dr. Kadi, Dr. Kajiduni, sir, Dr. Alagwadi, sir, and Dr. Brahma Prakash and T.K.S. Gowda are here. I welcome warmly all the previous former officers and the students of T.K.R. Reddy as well as R.B. Patil, sir. So warm welcome to you all, sir. And our officer colleagues are also here with us in this function. And I welcome warmly all the officers of the university. And in addition to that, many of the faculty of the various departments in the Agriculture College, Dharwad are also here. So I welcome each one of you. And always our strength is the students, and the PG students and the undergraduate students are over here. And I welcome all the students for this function. And with their presence, we could able to make many of the functions happen in the campus. So I welcome all the PG students. So in addition to that, the media and other persons are also here. So I welcome warmly all the persons who are participating in this program. And thanks a lot for giving me this opportunity to welcome the dignitaries on the dais. Thank you.
Thank you for welcoming the gathering and setting a meaningful tone for the proceedings, sir. On behalf of everyone, I request Dr. C.R. Patil, sir, to welcome Dr. V.B. Kuligod, sir, for this gathering. As our gathering begins to take shape, we now move to a key moment of the program. I would like to invite Dr. P.U. Krishna, sir, Director of Research, UIS Dharwad, to introduce our today's chief guest, whose journey and contributions continue to inspire us.
Dignitaries on the dais, colleagues, dear student friends. A couple of months now, it's been very exciting for all of us at the Department of Microbiology in anticipation of this day. And to be with us on this day and to deliver the memorial lectures in the memory of two stalwarts of our university who have developed the Department of Agricultural Microbiology and brought it to the extent that it is there right now. We have with us a leading soil microbiologist of the country. I have known him for the past 35 years and has been mentoring me at various levels for various purposes. So I do have an extreme privilege, an extreme honor of introducing him to this august audience. Today he'll be talking on the reflections of biological nitrogen fixation. A distinguished scientist with about 4 decades of research and development and experience in sustainable agriculture, focusing on biological nitrogen fixation, biofertilizers, organics, and soil health improvement. He worked in the ICR as project coordinator at the Indian Institute of Soil Science, Bhopal, where he headed major national and international projects on BNF. He also headed at the same time the AICRP on biological nitrogen fixation, which were later christened as the All India Network Project on Soil Biodiversity and Biofertilizers for nearly 2 decades. Later, on retirement, he was the ICR Emeritus Scientist, and during this time, he established the India-UK Nitrogen Fixation Center, also referred to as IUNFC, in collaboration with Oxford University. Dr. Rao graduated from the University of Delhi and earned his master's and PhD in microbiology. I'll digress a little bit to tell you that he joined agriculture, leaving behind a very interesting offer from AIMS to be a biochemist. And he continued to be in agriculture, leaving an extremely covetable position at the Tata Institute of Fundamental Research. He did his postdoctoral research at the University of Kent, UK, and was a visiting research fellow at the University of London, and the University of Sussex. He has more than 160 scientific publications in very highly reputed international and national journals, a lot of book chapters and conference papers. He edited quite a number of journals and is an active reviewer in many national and international scientific journals and funding bodies. Several awards and honors have been bestowed on him. He is also the fellow of the Indian National Academy of Agriculture Sciences and Indian Society of Soil Science. Recently, he has been nominated as a member of the Executive Council of the NAS from the year 2026. He has played a very crucial role in perspective planning of the ICR institutes and has been an advisory member in several Indian research institutes and national committees on soil health and biofertilizers. He has traveled very, very extensively and lectured widely in India and abroad as an invited speaker. He continues to mentor youngsters at various levels and takes keen interest in the history and philosophy of science. Today, let me have the honor, along all of you, to present Dr. D.L.N. Rao.
Thank you, sir, for the balanced and beautifully timed introduction of our today's chief guest. Just as one tiny microbe can initiate a thriving culture, a single flame can light up the entire space. We now come to the moment that symbolizes inspiration, growth, and illumination, the lamplighting ceremony to formally mark the beginning. Deepo Gnanasya Rupaka, Gnanena Prakashate Lokaha. I request all the dignitaries to lit up the flame of knowledge and offer floral tributes to our two great stalwarts. I kindly request all the students and faculties to come over the stage and offer the floral tribute. With immense gratitude, I thank all the dignitaries for lighting up the flame of knowledge. As we continue our program, we now come to a segment that celebrates passion, perseverance, and discovery. Through a rich tapestry of memories, we are about to glimpse the life sketches of two extraordinary pioneers of microbiology department whose curiosity and brilliance have left an indelible mark on their next generations. So without wasting a minute, let us step back, immerse ourselves, and allow this story of passion, perseverance, and discovery to unfold before our eyes.
Stand here? No, I don't require this. Thank you. I'll fall. Yeah. Sorry. Is it okay? Yeah, thank you. Has it opened? Yes, open. Yeah, you can just. You can read directly from the. You can do that. Will it take time? 5 minutes. Okay. 5 minutes is enough time. Very good morning, ladies and gentlemen. First of all, thank you very much, Dr. Patil, and all others for inviting me here. And more than the lecture, it's really a personal pleasure for me to meet so many old friends. And this is a very special occasion for me to meet all my old friends, Dr. Kulkarni, Dr. Bhagiraj, and all others. And it's just like having a Dharwad peda. It's very sweet. And it's wonderful, absolutely. So that's what I told the Vice Chancellor in the morning. As far as I'm concerned, the lecture is over because I've met everybody.
Sorry to
Yes.
interrupt.
Yeah.
There are some reminiscence of the two Star
Yeah,
Wars.
so let's start with that. Let's start with that, and then we'll start with the lecture later on. Yeah.
the video ends, we are left with luminous legacy of two remarkable scientists. Now I would like to call Dr. J.H. Kulkarni sir to share his thoughts and to share his thoughts.
Good morning to you all. I'm very happy we are holding the second memorial lecture. Our good friend Dr. D.L.N. Rao, who has been doing excellent work in microbiology at the national level, being a student of IRI, he has done extremely good work and is a good friend of all microbiologists right away in Dharwad, in Bangalore, and so many places. His work has been tremendous. I feel happy that he is the right person to deliver this second memorial lecture. I invite him also on my own behalf, and others also. There are our Bangalore colleagues with whom I worked, Dr. Bhagiraj, Dr. Siddaramegowda, and then Dr. Balasubramanian, and so many people. So I wish the function well. Thank you.
Thank you, sir, for your kind words. Now I would like to request Dr. A.R. Alagavadi sir to speak few words on this occasion.
Good morning, everybody. I had a very good time with Dr. D.L.N. Rao. We had a lot of interactions because I did my PhD at IRI. From that time onwards, we have been very close association because he used to visit quite frequently to our department. And afterwards, this interaction continued when I became the one of the member in National Bureau of Agriculture Important Microorganisms in 2 projects, and he was also leading many of the projects. So that's how for almost 8-10 years we were together in those projects. I'm very happy that he made it to Dharwad and is talking to all the students, microbiology students, and I wish him a very healthy and long life. And I also wish the function a grand success. Thank you.
Thank you, sir, for your valuable words. Now I kindly request Dr. D.J. Bhagyaraj sir to express his thoughts.
Good morning, everyone. And regarding Dr. Reddy, I know not much about him. And only thing I used to know that he was appointed as professor here at Dharwad campus. And when I came to Dharwad for something, for exam or something like that, then we had very good interaction. And he's a good microbiologist, that much I can say. And one question he asked me, can you speak Telugu? Then I said, I can speak Bangalore Telugu, not Andhra Telugu. Then he was very happy. Little Telugu we used to converse. And regarding Dr. R.B. Patil, I was assistant professor of microbiology at UAS JKVK. At the time it was Hebbal for some time, then JKVK. And Dr. Patil returned from US after completing his PhD, and he joined us associate professor of microbiology. We used to sit in the same room, okay? And he had a habit, the table and the chair, he will sit in the chair, put both the legs on the table. And somehow he used to tell me, sir, we don't do it here. Then he said, no, I am used to this in doing it in US. So that was there. And another thing was, I was also a part-time PhD student. And I completed all the courses in microbiology excepting fundamentals of microbiology, which is the first course. And so, but I have to finish another 3 credits. So Professor Rangaswamy told, why don't you take fundamentals at the last? So who offered the course? It was Dr. R.B. Patil. And the way you used to teach was wonderful. I can still recollect. And next lecture, he will be on genetics of microorganisms. He will tell pages 92 to 100 from Pelzar book. T.D. Brock book, this page to that page. So you read and come. I will take the lecture next time. So we used to read and come, and it was much easier for us to follow the lecture. So that's a wonderful thing he learned. Another thing was, you used to give the question paper, midterm test, give the question paper, and he will say, take it home. And next class, that is after 2 or 3 days, come back with your answer book, and then give your answer book. I tell you, that was more difficult than writing the answers as they give the questions. It was very, very difficult, very tough. So we learned so many things from him. So a great microbiologist and a wonderful person, both he and his wife. I enjoyed going to their house, and they have come to my place. So it's very happy to be here in this occasion where we have this memorial lecture. Thank you all very much.
Thank you for sharing your thoughts, sir. Now I would like to call upon Dr. Balasubramaniam sir to express his views.
Good morning, everybody. I happened to work with Dr. R.B. Patil for my PhD program. That is from 1973 to 76. When in those days, guides used to select students. But in my case, what happened, I wanted to do PhD under Dr. Patil. MSc I did under Dr. J. Rangaswamy, and when PhD thing came, I went and met Dr. Patil. I want to register under your guidance. I don't know, suddenly he agreed, and my colleague said, how is that you selected Dr. Patil? It is not very easy to work under him. But somehow, I was fascinated and attracted with his discipline and dedicated service. When I registered, I was asked to work on pesticide degradation. I didn't know anything on pesticides and all that. Somehow when the subject was introduced to me, I had to undergo 3 months short-term course in Indian Institute of Science under Dr. Bhatt, head of the Department of Organic Chemistry. And then Nagendrappa was my co-guide in our university because he was also an organic chemist. That is how I was made to work under pesticide degradation. And this I worked with, at that time, I had undergone the experience of Dr. Sidramya Gowda, who worked for his master's degree under Dr. Patil. And he was working on pesticide degradation. That is how I knew many things and techniques. I still remember Dr. Gowda worked on aspergillus, on benlate degradation. And, and it was really wonderful to work under Dr. Patil. I learned many things in my life. And after completing doctorate, he told me, you take a PRS examination. I was not very keen, but still I appeared for the examination and I was selected. When the posting came, I was posted in the Central Institute for Research and Cotton Technology in Mumbai. I was very reluctant to go. I don't know, that year I was the only candidate selected and I had no other go. Dr. Patil saw to that I go and report in Mumbai. That is how I joined ICR service, though I was assistant professor in the Department of Microbiology at Hebbal campus. Of course, when I joined Mumbai also, he used to visit my institute many occasions because his brother was staying in Mumbai. And he invariably used to visit my institute and check whether I am working properly in the area of microbiology, because that my institute was a post-harvest technology institute, not a production institute. Our production institute is in Nagpur, that is Central Institute of Cotton Research. But still, as a challenge, I was forced to take up many projects in a post-harvest technology institute. I could work very efficiently under the guidance of those directors, Dr. Sundaram, Dr. Betrabat, Dr. Patil, many people there, Dr. Kande Parkar, who was my head of the department. Dr. Patil knew all these people. When Dr. Patil was, when he joined Northeast Institute after his retirement, he met my director there, Dr. Sundaram. Yeah. And he told many things about me there. And that is how I continue to receive his guidance almost till his death. We celebrated his 75th year birthday also. I was fortunate to attend that program. And he was local guardian to my daughter, who did her dental course in Dharwad. And Dr. Patil was really an excellent human being, apart from his wonderful knowledge on microbiology. Thank you.
Thank you, sir, for sharing your thoughts. I request Dr. A.R. Alagavadi, sir, the first student of T.K.R. Reddy, sir, to brief his views.
I'm sorry, again I'm standing on the dais. Just say something about my teacher, Professor Ramchandra Reddy. I'm not the first student of Dr. Ramchandra Reddy. First student of Dr. Ramchandra Reddy was Dr. D. Radhakrishna and Mr. Furtado. Radhakrishna, of course, is then US Bangalore. He's retired. I just would say a few words, I read a few words about Professor Ramchandra Reddy. Dr. T.K.R. means Timojapalli Krishna Reddy Ramchandra Reddy. He He was born on 15 June 1929 in the village Kanipapam of Chittoor district of Andhra Pradesh. He had his early education at Chittoor, then completed bachelor's degree in agriculture from Andhra University, Faculty of Agriculture, Walter, Andhra Pradesh in 1950. He obtained his master's degree in agriculture with specialization in microbiology and mycology at the prestigious Banaras Hindu University in 1956 with first class, first in the order of merit. Dr. Reddy joined University of Madras, Faculty of Science, for his doctoral degree in 1956 in the Department of Botany, where he worked on influence of trace elements and metal chelators and rhizosphere microflora of rice. He was awarded the degree of Doctor of Philosophy in 1960. Professor Ramchandra Reddy, his higher academic training in India and abroad includes a postdoctoral fellow in Madras University for 2 years, from 60 to 62, then postdoctoral fellow, National Research Council, Ottawa, Canada, for 1 year, 62 to 63, as research associate in the Department of Bacteriology, McGill University, Canada, from 63-64, as microbial geneticist, Department of Microbiology, School of Medicine, University of California, Los Angeles, USA, from 1964 to 1966. He worked as agricultural demonstrator, Madras Agriculture Department for 3 years from 1950 to 53, and as agriculture demonstrator in Andhra Pradesh, Andhra Agriculture Department 53-54, as a lecturer in botany, University of Madras for 2 years, then worked as assistant professor, Institute of Biochemistry, Frankfurt University, Frankfurt, West Germany, from 68 to 71. He joined as professor of agricultural microbiology at Dharwad campus of US Bangalore in 1973 and served for 16 years. Dr. Reddy joined the newly started Department of Agricultural Microbiology at Dharwad campus in 1973 and is responsible for developing the department from scratches single-handedly. Since he was the only PG teacher in the department then, he used to offer many courses including history of microbiology, microbiology techniques, fundamentals, soil microbiology, microbiology of air, water, sewage, antibiotics, physiology, genetics, and so on. In addition to PG courses, he used to offer UG courses also, as there were no regular teachers in the Department of Agricultural Microbiology then. On research front, Professor Ramchandra Reddy has guided 20 masters and 3 doctoral degree students, assigning them with various topics including rhizobiology, BNF, renewable energy, foodborne infections, environmental microbiology, seed microflora, etc. He is the person who wrote and got ARCRP, Center on Renewable Energy Sources, that's biogas technology, or renewable research technology as it is called now, at Dharwad campus in 1966-67, 76-77, which is continuing even today in the Department of Agricultural Microbiology. He also obtained a project from Kada, Malaprabha and Ghataprabha, Government of Karnataka, with which department has acquired large number of equipments. Dr. Reddy has published a good number of research papers on various aspects like radiation biology, because he worked on radiation biology at USA, then soil microbiology, medical microbiology, environmental pollution, BNF, food microbiology, biogas technology, and so on. Most of these papers were published in international journals. Professor Reddy also was a member of various professional bodies, including American Society of Microbiology, Canadian Society of Microbiology, Indian Science Congress Association, Association of Microbiologists of India, Environmental Meteorogenic Society of India, and so on. He also served as examiner for PhD degrees in several universities, and after a long, fruitful service of 16 years as professor and head of the Department of Agricultural Microbiology at Dharwad, Professor Ramchandra Reddy laid down his office on superannuation on 30th June 1989. After retirement, he settled at Tirupati, where his better half, Madam Dr. G. Parvati, was working as principal of SV Medical College, Tirupati. While working on radiation biology, probably he was exposed to some kind of radiation, the effect of which caused lung cancer to him. After suffering for some time, he breathed his last on 28th of April 2005. The memorial lecture series organized in his memory, as well as one more stalwart microbiologist, Dr. R.B. Patil. We wish this series, and specifically Professor Dr. D.L.N. Rao sir. Thank you. Thank you.
Thank you for sharing your thoughts, sir. Having listened to all these stalwarts, now it is my honor and privilege to invite our chief guest, Dr. D.L.N.R. Rao, sir, retired I.K.R. Emeritus Scientist, ex-Project Coordinator, AICRP BNF, AANP on Soil Biodiversity Biofertilizers, I.K.R. IISS Bhopal, to deliver his distinguished lecture as we step into this inspiring segment of our event. We are all ears, sir.
Thank you very much. It was so inspiring to listen to the contributions of both Dr. R.B. Patil and Dr. T.K.R. Reddy. And I would say that this university was very fortunate to have them as their founding fathers after their extensive experience abroad, not only at their PhD level and their postdoctoral level. And their work has already been mentioned by Dr. Alagwadi in detail, so I would not go further. But all I know is that they are researchers at Madras University, to which I will be referring Dr. T.K.R. Reddy, and the researchers on biogas and biological nitrogen fixation. And above all the researchers, it is the visionary outlook and sustained efforts. These are the two things which are required, an outlook and effort. And that's probably what has led to the establishment of one of the finest schools of agricultural microbiology in the country, at Bangalore, at Dharwad, which everybody knows, and which is reflected in the results of yesterday. Dr. C.R. Patil was showing me of the various competitive examinations and various others, where the students have been doing extremely well. So congratulations for that. Now, I have chosen this topic of reflections on biological nitrogen fixation research in India in the 20th century, early beginnings to post-oil crisis. Now, why I have chosen this topic is because of I know something about biological nitrogen fixation, having been working on this field for about nearly 5 decades. And the second thing is, when I mean reflections, I really don't mean my own personal opinion. What I mean is the opinion of many other people, which I will be sharing in this lecture. And those people happen to be microbial ecologists like me. I'm not a person from genetic engineering or biotechnology. I'm a soil microbiologist, I'm an ecologist. So that particular perspective will become clear as I speak. And the third reason why I have spoken is this is actually about history of science. This is a topic which is very, very dear to me. And I was just listening to Dr. Alagwadi saying that there was a course here on history of microbiology also. And why I think history of science is very important is something which you will see in the last slides at the end of my talk of Martin Alexander, whose book of soil microbiology, all of you know that. History of microbiology or history of any subject, as I said, biological nitrogen fixation is just an example I have chosen because I know something about biological nitrogen fixation. It can be history of any subject, whatever students were attending from various. It's very, very important for us to know the history in order to know what was going on in the minds of those people at that time, about 100 years ago, when they did not even know how nitrogen is assimilated by plants. What was their approach to experimentation? So it is the thoughts, it is the approach to experimentation, what were the kind of experiments they were doing, that will tell us something about the kind of efforts that we are making, or maybe they will help us in our efforts. So as I said, for a long time I was thinking of writing a lecture on this biological nitrogen fixation history. And since I happen to live abroad for a long time, so last year I was there for 3 months, spending time only on this. And then when Krishnaraj invited me, so I was so happy to come and speak about this particular topic only because this is at Dharwad. I think this, or Bangalore, one of the best places where I could have given this. So what I will be covering is the work that was done in India. How did the research on BNF start? And also some of the research that was done abroad. In those times, people used to take soil samples from here, or microorganisms from here, and then work abroad also, some classical work. I'll be covering those work which have been extensively cited. I have tried not to miss any of my topic. I have shared my lecture also with one of my mentors, Professor T.K.R, who lives now in Mangalore. He has conveyed his best wishes. He knows Dr. Patil very well for a very long time. He also conveyed his wishes yesterday. So I conveyed that to the family of entire Dharwad family here. Right, so can I have the first slide, please? Yeah. So I have already said why history, why it is important, why is history important? And I have chosen biological nitrogen fixation as an example. And what is the importance of biological nitrogen fixation? I think everybody knows that biological nitrogen fixation is the second most important biological process on this planet. And the first is obviously photosynthesis, and after that it is nitrogen. And the symbiosis, I'll be talking more about symbiosis, about rhizobial nitrogen fixation, which has also been used by various people, as they have already called it, as a refined parasite. And it's also sometimes people call it as a royal battle between the plant and the rhizobium. When the rhizobium actually tries to, the plant tries to actually evade the symbiosis. But the bacteria is now able to overcome that. And in fact, as you know, biological nitrogen fixation is an anaerobic process. And it happens to be an anaerobic process because plants in their defense mechanism produce a lot of acidity. They produce a lot of protons. And the bacteria has to do something with those protons. It has to handle those protons. So it says, all right, let me fix some nitrogen with those protons. And in that process, nitrogen gets fixed. Hydrogen is also evolved. Ammonia is also evolved. And then you have to do something with that ammonium also, because if there's too much of ammonium, the pH will be very high. So that ammonium then happens to be excreted back into the plant. So that's how nitrogen fixation happens. And then this is restricted only to bacteria. There is nothing on archaea, which are the organisms which fix nitrogen. Starting with the oldest ones is Methanobacterium and Methanococcus and other organisms. And biological nitrogen fixation is not something which is universally present in legumes. As you know that the papillonoid legumes, about 90% of them, they fix nitrogen. And the sesquipinoids, they absolutely avoid nitrogen fixation. It's only about 30% of them which can fix nitrogen. The mimosoid legumes, about 70% of them, they can fix nitrogen. And it was something because it was required at a certain point of time. There was no fixed nitrogen on the planet, so it was required. And it is something which evolved about 3 million years ago. And it has, if you look at the phylogenomics of nitrogen fixation, among the families of these various clades, which in the rosette clade where the nitrogen fixers are present, it is not as if that they acquired nitrogen fixation at various times, and it was also lost at various times also, because when there's no need for nitrogen fixation, there was no need. So they will just do away with nitrogen fixation because it's a very energy intensive process. It requires a lot of ATP and a lot of other things like that. So having said that, now let me see why in our case, particularly in the case of Indian agriculture, why it became very important is the oil crisis of the 1970s. And I think you heard all the stalwarts just now speak. And I'm not a stalwart, but I'm just junior among them. So during that period when we were, the period that we're talking about, in 1970s, the price of oil was ranging between $1.35 to $3 per barrel. And suddenly in 1 year, it rose from 3.38 to 6. So it's what is called as an oil shock. There was a price for about 4 times in 1 year. And over a period of 5-7 years, it went up 10 times. The price of chemical fertilizers went up very high. So there was then a need to substitute a part of the chemical fertilizers with biological nitrogen fixation. And that was the great interest at the time in biological nitrogen fixation. Let me go to the next slide. Okay, so this next slide. Yes. So I've divided my talk into the beginnings, I'm talking about how biological nitrogen fixation research started in India, and you'll be surprised to know that some of you might already know that it started with a plant called as the Indigo, not the Indigo airline, it is about the Indigo experiments. It started at 1902. These were the Indigo experiments. Indigo is a nitrogen fixing legume, as you know. And the experiments were done between 1902 to 1910. And then the IARI, the Imperial Agriculture Research Institute, was established in 1905. And the researches that were done from 1910 to 1936, and then later on when it was shifted after the earthquake to Delhi, 1936 to 1950 at Delhi, and then the rest of India, what was happening in the rest of the country, post-independence, what was happening. And then I've chosen 1960 as a sort of a demarcating line, because after 1960, all the other agricultural universities and other institutions happened, and there was a lot of institutional expansion at that time. And then the post-oil crisis after 1975. I would stop somewhere around 1975, 1980, because after that period, a lot of information is available on the internet. A lot of reviews have been written. I have myself written 2 reviews in the Indian National Science Academy. Those are available. So I would not be spending too much time on that in the new millennium. Mostly the earlier work is not available on the internet. It's available only in the annual reports of very old institutes, as I said, at IARI, which are not very easily accessible for everybody. But I had an opportunity to go through all about 50 years of annual reports. So I'm going to share that work. But before I do about India, because there are students here and they should know how the biological nitrogen fixation as a process was discovered. I think everybody knows the names of Hermann Hellriegel and Hermann Wilfarth. Hellriegel and Wilfarth's name, I think everybody knows, 1888. They are the discoverers of nitrogen fixation at a place called as Bernburg in Germany. As you can see those pot experiments on the right here. These are the pot experiments on the right that you see. This is the glass house in which the experiment was done, and they used to have these trains in which you can actually put the pots inside and out. Now, why did they succeed? I just now said that the purpose of my talk is not to say that you can read in the textbook. But why did they succeed, and why did others fail? Others were also working. At that time, you must have heard the names of Boussingault, the French chemist, Laws and Gilbert, the people who started the Rothamsted experimental station, and Liebig, who's the father of agricultural chemistry in Germany, everybody knows his name. All these people were working on nitrogen fixation. At that time, there was a doubt about how does a plant get its nitrogen. Does it get it from the air? Does it get it as ammonia? Does it get it from the soil? And so on and so forth, those questions. In fact, on September 20th, 1886, when Hermann Hellriegel read that paper in Germany, the title of that paper was, "What is the source of nitrogen for the plant?" And why did he succeed while others failed? Is because the others, in trying to maintain a very sterile system, they were excluding dust, they were using steam to heat the air that was going through the experiments and all that. So in that process, they killed the microorganisms. Whereas what he did was to actually use a soil suspension and then keep the microorganisms intact. And that was the reason why they succeeded, whereas others failed. So this is a plant called as Serradella. This is a plant which is called as Erbsen in German, which means actually peas. And this is lupins. So this is, as you can see, these pots are uninoculated and these are inoculated. These pots at the bottom are uninoculated and these are inoculated. And he did another very smart thing. He used 2 different cylinders, as you can see side by side, in which a root of, this is the pea plant, one root was put into this, which is sterile, and another one is in which it is not sterile, in which a suspension of roots was kept. Elegant experimentation, this is 1886, and they found nodules on this. And even a bell jar like this, a glass bell jar like this, in which the quartz sand is at the bottom and the plant is growing, and the sterile air is forced through, but an inoculum is put. So I think we'll briefly stop here, just to give you a flavor of how the biological nitrogen fixation was actually discovered This is a paper which is in German. You can't get it in English. There is no English translation available. So at that time, as you know, Bayrink had already discovered Azotobacter, crucocum from soil and from canal water, and the first aerobic nitrogen fixing bacteria to be isolated was by Bayrink was in Rhizobium. Now, you would like to know who was the first Indian to actually go abroad, or who was the first Indian to actually work with microorganisms. When I mean microorganisms, I mean agriculturally important microorganisms, not the others in veterinary sciences and others. I'm just talking about agriculturally important microorganisms. So it was Dr. N.K. Pillai. I don't know how many of you have heard the name. He was from Kerala, then he was working in the Department of Agriculture later on. He worked with Lonis at Leipzig. And at that time, people were thinking that Bacillus also fixes nitrogen. So there was a claim that Bacillus subtilis and Bacillus megatherium fix nitrogen, but they found that these are unfounded. But two species of Bacillus mesentericus group, which is Bacillus malabarensis, was taken from here, from the Western Ghats, and Bacillus danicus. These were found to be capable of nitrogen. And then about Klebsiella, they were not very, very sure. They proved to be intermediate forms of bacteria pneumonia, azotoscore, and aerogenes. So he reported the results of nitrogen fixation. So Lonis and Pillai's papers are very classical papers on nitrogen fixation. And later on, Perry Wilson at Wisconsin also worked on Klebsiella. So let's move further to the next slide. So, as I said, now come back to India. We're now done with the European connection. Let's come to the Indigo connection. What was the reason why the Indian Agriculture Research Institute, or the Imperial Agriculture Research Institute, was cited at Pusa, in fact? And the reason was that indigo is a nitrogen-fixing plant. Indigo is a source of the dye, indigo, the natural indigo that you know very well. And indigo had been grown in India right from 18th century. And it was grown in Bengal, in various places in Bengal, like Barakpur and Nadia and other places. And then, because of, after the mutiny of 1857 and various other reasons, there were problems of waterlogging and nodulation is inhibited. That shifted from Bengal to Bihar. And in Bihar, I'm sure some of you have already gone to Pusa, Bihar. If you have gone there, within a 100-kilometer radius, these indigo stations were established. So one was a Bihar Indigo Planters Association. This is in Champaran district in Pipra. And there's an indigo improvement syndicate at Dal Singh Sarai. So the first bacteriologist, as they used to be called at that time, to be appointed in India was Cyril Jonas Bergtheil. He was a chemist and a bacteriologist trained in Nuremberg and Y College. And he worked there in 1902, 1903. But why did he work on indigo? Not on nitrogen fixation. Because the process of fermentation to produce the indigo dye, whereby the indigo tin is, indican is converted into indican, which is a dye, is a fermentation process. He was working on fermentation technology to improve. But at that time, what happened was in 1897, the Germans had succeeded in synthesizing natural indigo. So once the natural indigo became available very cheaply and in very large quantities, there was no market for the natural indigo that is produced here. So the indigo production fell, the prices fell almost to 15%. And it was then that these indigo planters decided to hire microbiologists to improve the fermentation process, to improve the indigo yield, as well as to look at how agricultural interventions could improve the yield, whether it's against diseases or waterlogging or maybe nitrogen fixation in that case. So that's how actually nitrogen fixation research started in India with indigo plant. And then in 1905, he was appointed as the imperial bacteriologist. In 1905, as you know, the Pusa Institute opened. There was an imperial entomologist, mycologist, economic botanist, agricultural chemist, Dr. Walter Leather was there, whose name everybody knows. But the imperial bacteriologist that was appointed was Cyril Jonas Bergtheil, but he started, but no work on Pusa was started. He was seconded to Sirsai, and then he first did what is called as a soil inoculation technique in 1907. So this was the first time that an inoculation experiment had taken in India, whereby soil from existing indigo plantations was taken and then it was put into other fields and then plowed into the fields and then look at how improvement of nodulation. And then nitrobacterine, as this was then called, rhizobium which was isolated. I will show you a slide also. But this rhizobium was not isolated in India. This was isolated in London by Dr. Bottomley of King's College. That nitrobacterine was then brought to India, and the first inoculation experiment took place in 1909. I will show you the results of that experiment. So, since Cyrus General Patil did not join at Pusa, Claude Mackenzie Hutchinson, so he was the imperial bacteriologist, who was appointed in 1909 at 1926. So you can say agricultural microbiology proper started in 1909 at Pusa Institute in Bihar. And he was then assisted by another microbiologist called as J.H. Walton. He was called as a supernumerary bacteriologist, and that is another bacteriologist. He was working on non-symbiotic nitrogen fixation, whereas Claude Mackenzie Hutchinson was working on symbiotic nitrogen fixation. And then he was assisted by one Dr. N.V. Joshi. In fact, N.V. Joshi is the first Indian microbiologist. These are Britishers. The first Indian microbiologist to have worked on microbiology is N.V. Joshi, Narayan Vishnu Joshi, about which I'll be talking later on. He was there until 1936. This is his picture. And then K.S. Vishwanatham, C.S. Ramaiah, and they were their assistants who were working along with them. And later on, Dr. S.V. Desai and Dr. N.D. Vyas. So these are the people who worked before independence, and I will shortly describe their work. Next slide. So this was the experiment. This is the report of the Indigo Research Station in Sirsaya in Bihar, which is about 100 kilometers north of Bihar, north of Pusa now. This was the first experiment done in 1909 and 1910. This was obtained from Professor Bottomley. And 1/3 of an acre was kept as a control, and 1/3 of an acre was treated. It's a very big experiment. We are not using that size experiments even today. And this is the weight of the plants in mounds, and what he writes is that there was an increase in these due to the rhizobium inoculation, but there was a reduction in the average indicon content, as a result of which there was not much difference overall in the indigo that was obtained from this. So from this he concluded that this is no good, that you can't have a culture from abroad and then have it work here. You'll have to isolate organisms from here. If I now tell you a summary of the entire experiment, this summary tells you the entire summary of all the experiments that were done at Pusa. They worked on microbiological activity and soil fertility, taking microbial counts. There was an azotobacter plaque test to test for nutrient deficiency. And then they worked on non-symbiotic nitrogen fixation by azotobacter, cross-nodulation and root nodule bacteria in 1920. This is a classic paper of Joshi, which everyone should read. And even PGPR, plant growth promotion by non-nodulating rhizobia. That rhizobia, when they are not nodulating, they are improving the plant growth, what is called as PGPR today. This was already reported. Blue-green algae nitrogen fixation, I'll be talking about that. And nitrogen fixation in the maize rhizosphere. And they said there are endophytes in maize which fix nitrogen, even then, those papers which you have read. And the importance of micronutrients like copper in boosting nitrogen fixation. And others like decomposition of green manure, cellulose decomposing bacteria, and others. So let's quickly now look at the results of that. And this is something which actually blew me away when I read these results about 10 years ago only. In 1910, they were looking at the occurrence and activity of bacteria at varying depths in the soil, sampled up to 9 feet. In my life, I have never taken a sample beyond 90 centimeters. Beyond 90 centimeters, I have never ever measured microbiological activity in any soil, but they had measured up to 90 feet and found numbers of even 10 to the power of 5 bacterial numbers at 9 feet. And this is a very important statement which Hutchinson made, which I strongly believe. If Dr. Kulkarni may remember, 1985, Dr. Bhagiraj, you were all at Hisar at a symposium. At the time I was talking about the uselessness of counting microbial counts, that you only look at microbial biomass or carbon dioxide evolution and things like that. Hutchinson in 1913 made this statement. Kindly listen to this statement. The method of determining biological activity by plate counts has been abandoned in favor of measurement of the physiological activity of soil microorganisms and using carbon dioxide evolution, the optimum moisture content for bacterial activity in soil was determined to vary from 16 to 37% in different soils. I think I don't have to speak anything further on this. This statement itself is telling you everything. In 1916 and 1917, they reported defective nitrification in ranchy soils, which is improved with nitrifying organisms or lime or oil cakes as inoculum. Even today, Dr. Saab, liming is the standard treatment for treating of acid soils, improves microbial activity, and nitrification is, after all, the most sensitive, if there's one parameter, if I have to look for as indicating a soil health activity, it's obviously nitrification, the most sensitive process. And they said that it's only putting lime or oil cakes, or even they were inoculating nitrifying organisms. But one very important statement has made Dr. Alagwadi. He says, a very important observation on phosphate solubilization. The results so far do not suggest that solubilization of tricalcic soil phosphates by bacterial action is sufficient in amount to produce a surplus supply of soluble phosphate for the direct use of higher plants, but rather that the bacteria dissolve only sufficient for their own requirements. Whatever phosphorus they are solubilizing is for their own requirements, but in the process there is a boost of microbial activity, which in general then leads to, since nitrification requires phosphate, the carbonic acid resulting from bacterial action will in the soil in course of time solubilize sensible quantities of mineral phosphate. He is not discounting that microorganisms solubilize phosphate. But what he is saying is that the phosphorus that is solubilized by microorganisms is sufficient for their own activity, and that in turn boosts microbial activity overall, which in turn solubilizes phosphate, whether it's from calcium carbonate or from any others in the rhizosphere. And that's a very important distinction which is made, and a lot of people had been actually saying these things, but this had been said a long, long time ago. And that's why I said we need to know from history what has already been written. Now we come to symbiotic nitrogen fixation. Cross-inoculation of rhizobia, Joshi, as I said, Dr. N.V. Joshi, as I just mentioned, he recognized that CISR is a separate group, that it doesn't cross-nodulate with any others. This is something which, when we classified rhizobium, I think it's only in the mid-80s we started saying CISR rhizobium is different, therefore it is not rhizobium. It is not bradyrhizobium. Now we should call it by a different name. We started calling it as Mesorhizobium ciceroe. But look at the work of Joshi in 1915. And then, this is the first thing, that cicer is a separate group. And then he says rhizobium inoculation exercised a stimulating effect, even if they did not nodulate a plant. Look at that statement. He says, and his experimental data is a paper which is about 200 pages pamphlet. Substantial increase in root and shoot growth were observed even though there was no nodulation. An elegant experimentation which I will now show after this. Especially when cultures from one plant species were inoculated on a different species. That means when he was using cicer rhizobia on a different cross-inoculation group, he was getting a greater effect. And then he also said that there was a stimulation of plant growth observed with azotobacter inoculation and growth of legume bacteria isolates in artificial media separated by porous cylinder from the soil containing the growing plants. Elegant experimentation, as I said. Legumes, they used peas and mott, and non-legume oats. So what they did was, they took pots, they put soil, the inoculated microorganisms. But in another pot, what they did was, they put a porous cylinder in between the pot, put the rhizobium inside, so that the rhizobium is not directly in contact with the soil. And then obviously some of the rhizobia would migrate, and then to see if there's no direct contact between the microbe and the plant roots, what happens to that? I will show these pictures later on in the next slide. So these are the things. If you see this now, this is oats, Avena sativa, cereal. These are experiments of Joshi in 1919. And since this is very small and you cannot read, I will read them. This is the control oats where nothing has been inoculated. This is where potassium nitrate has been applied. So there's you can see a huge spurt in growth. This is where azotobacter has been applied. And this is where rhizobium has been applied, 3 different rhizobium isolates. Okay? And what are these rhizobia? These are peas, these are fascioles, and this is Sisarga. So this is rhizobium inoculation behaving better than azotobacter on oats. And this is the porous cylinder. In this spot, what they had done is they had a porous cylinder in the center, and then the rhizobia they had inoculated in the center, and then you can still see a stimulation. And if you look at the roots of those, this is control, this is potassium nitrate obviously, this is azotobacter, and this is rhizobium stimulation of the roots. And this is the one which is a porous spot. I'll go back to the previous slide now. And they also made a modified medium, soil extract, mannate, asparagine, agar was found to allow ready isolation of the organism directly from soil. Not only azotobacter, but they could also isolate rhizobium directly from the soil, something which Bromfield did later on in Rothamsted in 1980s. He found that specific medium for rhizobium. But they had given that medium there. And this is not appearing in any of our textbooks. And then there was a conference of chemists and bacteriologists at Pusa in February 1919. This is where these results were reported, and this is undoubtedly the first conference on agriculture microbiology in India. And there was a resolution on BNF there. They made a resolution that, that because India is lacking in nitrogen fertilizers, we need to improve, research on this. So this is that memoirs of the Department of Agriculture in India. This is Joshi's book. And then he found that chickpea, this is chickpea, as you can see, inoculated with cowpea and her rhizobia, it did not nodulate. That is where he's showing specifically that there was no improvement, that there is an improvement in the root growth with inoculation, but there is no nodulation. This is absolutely mind-blowing work. As far as I'm concerned, I don't know why this has not been more advertised. Once this, this paper is published, then I think people will come to know about this. We didn't notice that work, but people internationally have noticed that work. What did Ellen and Ellen say? Ellen and Elizabeth, Ellen and O and Ellen and Ian Ellen, I think you know the microbiologists in USA. They mentioned that Ferrer and Barker and Friesner in 1926 have also reported that root nodule bacteria might benefit plant growth without forming nodules. They noted, what did Ellen and Ellen say? Joshi was probably the first to suggest that root nodule bacteria existed in the tissues of certain papilionaceous plants, although root nodules were not formed. Let us put this in perspective. Now this is my sentence. This is my interpretation of that. Let's put this in perspective. Joshi never claimed to have found rhizobia inside the roots of non-nodulated plants. He clearly stated that rhizobia were found on the roots, but did not penetrate. And further, what did he say? A search was therefore made by cutting sections of such roots after sterilizing the outside. Although many sections of the roots were examined, no organisms could be detected in them. The possibility of finding the nodule-forming organisms inside the root tissue in the complete absence of nodules on the roots is a very remote one. So what he's saying is that if nodules are formed, we know about endophytes being there. But if nodules are not formed, the chances of finding roots, rhizobia, inside the roots is very remote. Now, I think I stopped there with symbiotic nitrogen fixation. Now let's get to non-symbiotic nitrogen fixation. Walton, as I said, he's a supernumerary bacteriologist, and he said that there's increased nitrogen fixation in soils where algae were growing. They were estimating nitrogen in the soil, and they found that there's increased nitrogen fixation where algae were growing. And then this was mentioned by Hutchinson later on, 5 years later. In the 1927-28 report, Joshi mentions, by alterations introduced in the experimental technique, I have made it certain that algae do fix a certain amount of nitrogen by themselves alone, as any possibility of symbiotic action with bacteria was prevented. Perhaps algae contribute a large share of the rise in nitrogen content in soils observed towards June or July, that is up to the break of monsoon. Why am I making these statements? Because at that time, people were thinking, does bacteria fix nitrogen? Is it the bacteria associated with blue-green algae in the mucilage, as you know, does it fix nitrogen? Or is it that algae are fixing nitrogen? Do they fix nitrogen? So there was a controversy about that. There was a lot of people. It was only in Drews in 1928 who said that, yes, he obtained blue-green algae. He said it fixes nitrogen. And then in 1936, P.K. Day is Pran Kumar Day, as you know, his work was published in Philosophical Transactions of the Royal Society of London. That was the work that he did in London in Queen Elizabeth College. He was working in Bengal at the time in Dhaka. And he found that in the rice season, there's a lot of nitrogen fixation in the rice fields. But still, nobody had obtained bacteria-free cultures of blue-green algae to prove that nitrogen fixation occurs. It was P.K. Day was the first one to do that in the world. The 1939 paper is a very classic paper, which again should be read by students, and I'll just refer to that. I'll come to that later because we are skipping that period. There was another very important paper. This is also a very favorite of mine, and all soil scientists should again read this. You know, soil scientists at Ludhiana and various other places, people also say that when you're growing a legume, and then you're growing a cereal after that. Okay, a green manure like mung, followed by rice, or any other green manure followed by a cereal plant. And they always say you should put phosphorus to the legume, and not to the cereal that is following. Interaction, I want you to pay very close because they are very precise in writing these things, you know. Those people, they were very well trained in writing the things. Each and every word carries a meaning because here we tend to write too much. The role of soil bacteria in provisioning soil phosphate in the form of organic phosphorus compounds because of the interaction of green manure and other decomposing organic material in the soil and phosphate manure such as superphosphate was attributed as responsible for the remarkable influence of green manure on the fertilizing power of superphosphate. The green manuring is improving the fertilizing power of superphosphate. Now, and this is italics are mine. We now know the importance of microbial biomass phosphorus in supplying a steady source of phosphorus to the plants. Yesterday I was visiting your organic farming division, and that's what I was telling them, that they should measure microbial biomass phosphorus in the soil and how it changes over time. That will give you a good indicator of how the phosphate nutrition of the plants is happening. Before I come to P.K. Day's work, because that's after a certain period, I just want to digress because we are now on organic farming. Albert Howard, I think everybody knows his name. There's no need to introduce Albert Howard. Albert Howard worked at PUSA from 1905 to 1924. He was an imperial economic botanist. He was a plant breeder, as you know, and Howard varieties of wheat, you have all been taught about that. But he was also interested in working on composting and organic manuring, things like that. So recycling of organic residues and composting. And you all know about the indoor method of composting, about the Bangalore method of composting, the two methods that you are normally taught, Bangalore method and the indoor method. He developed the indoor method, but then he was in PUSA. You'll ask me, how did he develop the indoor method if he's at PUSA? So I'm just coming to explain that. What did he write? He's a plant breeder. What he observed was that the farmers who are growing outside PUSA, their crops, they are coming, their crops are better than some of the crops which are growing inside the farm. And they were using more organics, obviously. So he writes full possibilities of the improvement of a crop variety. This is a plant breeder writing, right? Not a soil scientist. He says full possibilities of the improvement of a crop variety can only be achieved when the soil in which the new types are grown is provided with an adequate quantity of organic matter, humus. Plant breeding, to achieve any permanent success, would have to include a continuous addition to the humus content of the small fields of the Indian cultivators. There is another reason why humus is important. It is essential for the proper functioning of the second contact between the soil and the plant, the mycorrhizal relationship. What is the first contact between the soil and the plant? The roots. Obviously, the roots are the first contact between the plants and the soil. And this is the second contact. He's not a microbiologist. He's not a biologist. He's an economic botanist. It would follow that on the efficiency of the mycorrhizal association, the health and well-being of mankind must depend. And he, in fact, goes on to say that mycorrhizal is more important than biological fixation because some reasons. In the mycorrhizal association, nature has given us a mechanism far more important and far more universal than the nodules on the roots of the clover family. So he was a mycorrhizal proponent of organic farming, and then he said that this is going to be more beneficial, and we now know the relationship between mycorrhiza and symbiosis. Mycorrhiza evolved much, much before symbiosis did. There are common sim factors involved in all that. But what does he conclude? But interdisciplinary studies involving plant breeding, plant pathology, and soil fertility were not possible during those days at PUSA due to the organization of the discipline of agriculture into 6 separate sections. In those days, it was watertight compartments. So soil science department is working on composting, so Albert Howard cannot work. So therefore, he actually moved out in 1924 from there to Indore, only because he was not allowed to work. And then he went to Indore and then started the Institute of Plant Industry, where the Holkers had given him that place. And it's still there in the Indore College. If you visit the Indore College, you'll see Albert Howard's laboratory even today there. Now, again, this is actually, although it says Hutchinson 1925, that he was the in charge, head of the department, the section, he writes the report, but this is actually the work of Walton, where they found that this was the first time that non-symbiotic nitrogen fixation was estimated in the soil. Till that time, they were estimating only in what is now, we now do in Jensen's media. So it was a medium similar to that. So they were measuring symbiotic nitrogen fixation in the culture medium. This was the first time it was done in the soil, and this in a period of 1 month by adding mannite. Mannite is mannitol. They found nitrogen fixation was 3.5. But when you take Karnal soil, and because I worked at Karnal and have a weakness for that, so I picked up this slide. Karnal soil nitrogen fixation is 5 times that of Pusa soil. Okay, and when you add mannite, it is even much, 8 times. Why? They did not mention why, but I know why. Because they did not mention the pH of the soil. The pH of the soil at Karnal is never less than 8.2, more than that. And due to bacterial action, calcium is liberated, which is good for nitrogen fixation and bacterial growth. That's why you have more nitrogen fixation. They even discovered a new cellulose decomposing bacteria, which is a bacillus at the time. And then very important experiments, they recognized at that time, they said that this bacillus alone will not decompose cellulose. On its own, it will not decompose cellulose. It requires a companion of other organisms. That means your consortium. That means there are other things which are required to assist nitrogen fixation. It will not do it free of cost. Now this is Sen. This is J. Sen, Jananath Sen. He was a chemist. And he found out the same thing which Albert Howard had said, that the endophytic, he made one of the earliest suggestions that the nitrogen nutrition of a cereal crop could be met by the activity of the associated nitrogen fixing bacteria. So associative nitrogen fixation was discovered in India in 1929. And this is not there in our history books, not in Martin Alexander's book. Endophytic bacteria in rice roots of cultivars outside Pusa yielded slimy bacterial cultures of capsulated bacteria, microaerophilic. So you see, this is something very, very close to Azospirillum, although he did not name it as Azospirillum or did not even identify that organism. And he found the same results from endophytic cultures brought over from Agricultural College, Coimbatore. This is 1929, and nitrogen fixation was quite huge. It was almost 20 milligrams of nitrogen per liter under aerobic conditions. This is a work, you know, when I was doing my master's in IRA, there was a student from Thailand. His name was Eng Leong Phu, and he was working on Rhizobium phages at the time. And he took electron microscope pictures. And I thought that this was the first time in India that somebody is working on Rhizobium phages, and that was published in Current Science, that paper. S.V. Desai, root nodules of several leguminous plants and maize roots yielded bacteriophages. Bacterial numbers of 38 million per cc were reduced to 1,000 per cc in 24 hours. It was suspected that bacteriophage may have some connection with the benefit that gramineous plants directly derived from the culture of root nodule organisms. It is a very important statement. Kindly pay attention to this because I'm going to explain this. A few years ago, I was writing a book chapter, and somebody wrote a book chapter said that bacteriophages we should study in the soil because they create turnover of nutrients due to their latic action on the bacteria. And this is already written so nicely here. And I thought that was something there, we just missed this. Bacteriophage may have some connection with the benefit that graminaceous plants directly derive from the culture. That means when you are inoculating let's say a rhizobium on a cereal plant, it could be that the benefit that is coming, even though it is not nodulated, could be coming from the latic action of the bacteriophages also. This is something which we never ever even contemplated. So, Joshi isolated a pectin-producing facultative anaerobe from a maize-grown pot soil. And there's another scientist called N.D. Vyas, Narayan Dular Vyas. He again worked at IRI later on, after 36. The plates made from a juice of a one-day-old maize silage gave pure cultures of a raised, slimy, glistening colonies of Achromobacter fermentations. Kindly remember this Achromobacter, because I'm going to come back to work done during my time when I was a student on Achromobacter at IRI. I'm going to come back to that. Achromobacter, pointing to its endophytic nature. Root washings of 1-week-old maize stimulated nitrogen fixation by a mixture of nitrogen-fixing organisms grown from a soil inoculum by 15% over control. So the importance of maize root exudates in stimulating nitrogen fixation in the rhizosphere of croplands by over 50% was reported. And Dr. B. Vishwanath, I think every soil scientist knows him, Rai Bahadur B. Vishwanath was the first Indian director of the Indian Agriculture Research Institute, PUSA. Everybody who studied at IRI would see his photograph in the director section, B. Vishwanath. So at that, why he reported that on the bacteriology section in 1937? Because in 1934, the bacteriology section was closed because both Hutchinson had left, Walton had left, Joshi was an assistant microbiologist, and the section was closed and merged with the chemistry section. More critical experiments were carried out with organisms that survived washing the roots with mercury chloride. And organisms have been found associated with the roots of wheat, rice, and maize plants. Those associated with maize are still under investigation, but those associated with wheat and rice roots have been found to fix nitrogen. The amount of nitrogen fixed is of the same order as that of the leguminous organisms. This, along with the subsequent there were field experiments at IRI and endophytes in 1940. Proves the discovery of associated symbiotic nitrogen fixation in India much earlier than in the world literature that you're normally taught. Now, I think why I'm talking about is to tell you back about some international work done at the same time. P. N. Raju's work, about in Oregon, about the occurrence of nitrogen-fixing Enterobacter cloaca in the rhizosphere of maize. While he did not identify that organism, N.D. Vyas, it could well have been some organisms like this. And then about rice, there has always been a lot of discussion about, I did my PhD with Dr. G.S. Venkatraman, who was an algalogist, so he would claim that it is blue-green algae are responsible for nitrogen fixation in rice, and that bacterial nitrogen fixation is not so much. And if you cover with dark, then there will not be much nitrogen fixation. So acetylene reduction techniques and various other things like that. So there was heterotrophic, and then there was also chemotrophic fixation of nitrogen in rice reported by Dr. Gowda and Watanabe of your own university. The reason why I'm citing these works is that, remember that when there is nitrogen fixation, there is also hydrogen evolution. I just said that legumes, they produce a lot of protons and acidify the environment, so that is a plant defense mechanism. But the rhizobia have learned how to deal with their protons by fixing nitrogen. And some of them, as I said, acetylene, they reduce to ethylene. And some of them are so powerful that they can even reduce it beyond ethylene to even ethane also. So there's both chemotrophic fixation, there's also chemotrophic fixation. And there is a feeling that nitrogen fixation in rice is underestimated in global nitrogen budgets. I will be coming to the global nitrogen budgets as my very last slide, and some of that is underestimated. So you need more experiments to actually find out what is the nitrogen budget properly. Coming back to Joshi, the buried slide technique for direct observation of bacteria, they used buried slide technique, and Rossi and Collodny's, I have never used it, I think I have used it in my practical soil microbiology 101 by dipping it in the soils. During April, May, and June, he says Clostridium were predominant, especially after the rains or when moisture is added. So they were measuring the activity in 6 millimeters and 20 millimeters. Below the depth of 1/4 to 3 inches, that is 6 millimeters to 20 millimeters, hardly any azotobacter were present. Now, normally what happens is that we work along with soil scientists. They will take a sample of 0 to 15, 15 to 30, and we also measure microbial activity 0 to 15. We have never thought of actually taking millimeter sections of soil and studying. I have done that. I have taken sections 0 to 7 millimeters and 7 to 14 millimeters, 14 to 21. I have studied how the metabolites move through the soil, blue-green algae metabolites move through the soil. So this is also a statement which is worth examining, that it appears that the importance of the Clostridial group is far more important than the Azotobacter group, whose claims are perhaps exaggerated on account of its capacity of fixing large amounts of nitrogen in pure culture. So he says we should not now depend only on pure culture, see what's happening in the soil. And then they also did a microbiological test, determined the deficiency of PRK in both soils, called as bacillus. So that is the work that was done at PUSA, and it has taken nearly a long time. I'll have to now quickly run through the rest of the slides. So what NDVR said is that he did field experiments by saying that residual effects of sun hemp and wheat are more than that of maize, but not significant. He concludes that nitrogen fixation by the maize is due both to the stimulation of nitrogen-fixing organisms in the soil by the root secretions of the maize plant and by the maize plant itself. And then they did work on phages of rhizobium. Berseem is Trifolium alexandrinum. When they introduced it in Delhi, this is what is called as Berseem sickness. It would not grow. And then that was because of phages. And then they said that fenugreek or methi did not develop bacteriophages even in the 5th year. So the conclusion that they reached was that if you grow legumes continuously in a soil, then there is a chance of the building up of the bacteriophages. But if you rotate it with certain other crops, then the chance of bacteriophages developing, I'm talking about bacteriophages of rhizobium, is lessened. And this provides a beautiful explanation for the scientific basis of why we need crop rotation. That's why we need crop rotation already. And this is a picture of Dr. Narayan Vishnu Joshi, as I said. He has done so much of work on rhizobium, but not recognized. And the director of the Imperial Agricultural Institute, 1940, he wrote when he retired, one of the foundation officers of the institute, having joined immediately after its establishment, Mr. N.V. Joshi is a pioneer worker in the field of agriculture microbiology in India, and has made original and enduring contributions to the microbiology of Indian soils and to the subject of bacterial disease of crops. And it's my personal view is that he has a rightful claim to be the father of agriculture microbiology. So much of work that I have now told you. After retirement, he worked in College of Agriculture, Pune, then established the Agarkar Institute as a secretary in the Department of Microbiology at Pune, did a lot of other work. So let's come back to the rest of India. What was happening in the rest of the country in this period is that in the Madras state, the bacteriology wing was established in 1921 at Coimbatore. And then it was immediately closed and revived in 1929. I think everybody from Bangalore knows the name of Ronald Norris, needs no introduction. He was a government agricultural chemist. And then for about 6 years, and then he joined Indian Institute of Science as the head of the Department of Biochemistry. He did a lot of work on agricultural microbiology, metabolism of Azotobacter crucocum. And from here onwards, I will not be now speaking in detail because my part about the history of that part is over. Very quickly, I'll run through the rest of the slides because it contains a lot of valuable information on the history. P.K. Day, I've just mentioned to you, Pran Kumar Day in Dhaka, discovered Azotobacter indicum in 2 acid soils from Dhaka and Burma, and these are classified as Byrhynchia indica type species. So Byrhynchia was discovered in India. Byrhynchia was discovered in India, and so was Derksia. I'm just coming to that now. P.K. Day experiments at Dhaka and London. He was the first person to obtain bacteria-free cultures of Anabina. First person in the world to have actually made bacteria-free cultures, not using the traditional, because mercury chloride also, it survives. So he devised a special technique. We need not go into that, read the papers. And that was published in 20 important newspapers in England. When he made that, I think I still remember, if I can verbatim, I'll try to quote, Bengal Boy's Success or something like that was the headline. And these cultures were exhibited at the Empire Exhibition in Glasgow in 1938. And these were Anabina species, Anabina variabilis, Anabina gelatinosa, and naviculoids. And then on blue-green algae, I cannot leave without mentioning the famous botany schools at Madras and Varanasi. Desikachari's blue-green algae taxonomy was a Bible for us. All of us have used it. It used to be on my microscope all the time because look at every form and then identify it only from Desikachari's book. And then the schools at Varanasi, Dr. Arun Singh, his work, all these papers were published in Nature on reclamation of blue-green algae. And then there was K. Adinarayan Rao. He was a student from Mysore State. He was trained at Pusa in 1923. He reported leaf nodule bacteria in Pavetta indica and Chomelia asiatica, and that these were capable of fixing nitrogen. And then you can just read that. And then even seed-borne organisms, spermosphere organisms, this is at Indian Institute of Science by Sadhya and Subramanian. And Casuarina nodulation. Casuarina, again, there was a lot of confusion at that time. Is it nodulated by a bacteria? Is it nodulated by a fungus? Is it something which is beneficial at all? So, as a barber, he used to say that he has actually written that these outgrowths in Casuarina, these are disease causing and they should be removed. And in fact, in Andhra Pradesh, in Visakhapatnam district, when the forest department used to transplant Casuarina, they would remove those nodules, which is very beneficial because they thought that this is a disease causing. But it was Narasimhan who was working in Bangalore at the time. He was an assistant mycologist in Bangalore, and I think it says Agricultural Laboratory Bangalore. I assume it must be Hebbal at that time. So he observed nodules on in Madras coast and Bangalore, and then in 1918, he has produced a photomicrograph of the nodules. This is the photomicrograph, and this is a section of the nodules. 2.7 milligrams of 100 milligrams of nitrogen fixation was measured. And then Adinarayan Rao in another paper mentioned that it is 6.1. And there is one Chowdhury. He was working at Lahore, which was part of India at the time, undivided India. He used a crushed nodule inoculum to reproduce nodulation. But it was Aldrich Blake working at Dehradun in Indian Forest Research Institute that he was the first time he used a crushed nodule inoculum to increase it, and he showed that ammonium nitrate, this is control, ammonium nitrate can produce the root harvest 10 times, but if you inoculate it, you can get it 42 times the radial biomass. These are the first pictures ever taken of a Casuarina plant which has been inoculated with bacteria. They did not know that it is Frankia at the time. It was just a crushed nodule inoculum. They have not inoculated that. At that time, Layalpur, which is now called as Faisalabad in Punjab, that was a part of India at that time. They were working on nitrogen fixation, and Barsim, Lander, and Ali, they were doing inoculation studies in Punjab in 27, 29. They were distributing cultures at that time, 7,000, 8,000 cultures. Lakhs of 7,000, 8,000 cultures initially, but lakhs of cultures later on in Punjab on Barsim, which is Trifolium alexandrinum. And they used to produce the carrier material. Why I have written is because it's very interesting. We do it in polythene packets now. At that time, it used to do in cigarette tins. Yesterday at dinner, we were talking about olden days. In those days, cigarettes used to come in those cigarette tins. Like your pan masala today, it comes in those tins, exactly the same. So they would pack muck in that, sterilize them, and then they would inoculate that. And then Dr. Sannyas Raju was doing experiments at Coimbatore. They were all trained at Pusa at that time. They were working on legumes at that time. And Rajgopalan at 1930, he was the first to report on rhizobial denitrification. And the world acknowledges that. And the world acknowledges that whenever rhizobial denitrification is written, please read it. This paper you'll find is the first one that has been done on rhizobial denitrification. And then Raju, who was working at Coimbatore and Madison, he was working on effectiveness with radiation. And then in post-independence, as I said, azotobacter become very popular as indicators of soil fertility. Roma Bhattacharya, she was the one who actually isolated Dirksia. This paper was published in London, and they sent it to Dr. Waxman for identification. He said, I'm not the expert, you please send it to Jensen and Peterson, and then they identified that. So I just said, Barrinkia discovered in India, Dirksia was also discovered in India. And then these azotobacter and phosphobacter preparations, you know very well. They were brought from Russia. They were called azotobacter and phosphobacter. And in fact, the phosphobacter and azotobacter preparations were brought by the minister. At that time, there were no rules on bacterial. You don't need any clearance of the customs and all that. You just bring whatever you want. So they brought it to IARA, and then they were doing that. And then I think everybody knows the work of Dr. J.V. Bhatt. Sorry, J.B., not J.V. J.V. Bhatt. I'm sorry, there's a mistake in the slide there. Dr. J.V. Bhatt, Indian Institute of Science, Bangalore, he was a part of the BNO project, and he found a lot of azotobacter and earthworm casts. So when you're adding earthworm casts and all that in soil fertility, there's improvement of their quality. And then even soybean rhizobia You know, soybean rhizobium were brought over from America and introduced into India in the 1960s in large scale, as you know, 72,000 acres in Madhya Pradesh and other places. But at that time, soybean was an exotic crop for India at that time, but the strains of rhizobium were already present. And then the cross-inoculation groups of Sannyas Rajat established, then work at Pune and BD, and Annamala University, Dr. Rangaswamy's work, I think everybody knows. And this paper is very important, Rangaswamy and Subbaraja in 1962, because this is the first time, every time, we only know about Azotobacter crucocum, but this was the first time that they studied the Azotobacter species in wet and dry season of rice. They counted every 10 days for 9 months. So imagine how many observations were taken. 9 months into 36 samplings, and then from every soil to isolate and count and then do all the physiological tests, and found that the Azotobacter crucocum were the most predominant and efficient in nitrogen fixation, followed by Azotobacter indicus and Azotobacter lacticogenus, agilis. Berinkia and Vinelandi were less in numbers and less efficient. This paper should be read by people for those interested in history. And even, you remember, the sap exudates used to be done for ureide analysis for nitrogen fixation. I think when people were not doing N15, you were doing ureide analysis at one time. But these were being done much earlier in asparagine, glutamic acid, and glutamine. If they are there, it was found that the nodulation is effective, and if it is aspartic acid, it is ineffective. And then some work on cytology, stem nodules of Eichenomina, and my own supervisor, Dr. G.S. Venkatraman, he published this paper in Nature. In addition to the rhizobial bacteria, blue-green algae endophyte named Nostoc punctiforme occurred inside the nodules of berseem. So inside a root nodule of a legume, you have a blue-green alga inside that. And the first time in the world, this is a paper published in Nature, again, blue-green algae, DNA and RNA isolated from blue-green algae. And then, as I said, there was a lot of institutional expansion. I'll very quickly cover this, the divisions of microbiology at IERA and all the state agriculture universities, the ASERPs, the PL480, the AP assessment schemes, the Department of Science and Technology, the DBT. So much of work was done on Azotobacter at that time in the aerial roots of banyan, rhizosphere and phylosphere research at IERA and Indian Institute of Science. And then Achromobacter delmarvi, now I come back to that. This was one of the first organisms, along with Azotobacter, discovered on the phylosphere. And this was my master's work, where I showed that indols and gibberellins were produced by Azotobacter, and they increase the vitamin C content in tomato. And Dr. Shinde's work needs importance, because he was the first one at that time in India who said that Azotobacter not only fixes nitrogen and produces growth promoting compounds, but also controls plant diseases. So what we now call as PGPB. And then these experiments are done in front of my eyes, using Fusarium and Pythium liberanum, so many other funguses which are controlled with that. And then there are very few people who have used N15 analysis, and my friend Dr. Rajaram Mohan Ravi at IERA, at CRRI, and then he had a project from IAEA for a very long time measuring nitrogen fixation of about 5 to 10 grams, 5 to 10 kg in flooded soils and stimulated by rice straw application. And then this is very important, Dr. Lakshmi Kumari at IARI, she was the first one who isolated azospirillum in India. Again, this was done in front of our eyes. And that was sensational news. And then the growth promoting ability of BGA and vitamin C in tomato. And then the culture collections are very important for a microbiologist. You had the mycological culture collection in IARA in the division of pathology. But there were no bacterial collections at that time. The blue-green algae collections were there at IERA, my own supervisor, Dr. Venkatraman. And B.P. Pal's name, I think all of you would have heard. He was the first Director General of the ICER. And he had also worked on algae on T.K.R. He also had a collection, and Dr. M.S. Randhava was an ICS officer who was the Vice President of ICR. He had also worked on algae. He has also had an algal collection, and I have seen all these collections in my laboratory. And then Anabena azoli was an endophyte of Azolla, but never proved to have fixed nitrogen outside in the culture medium. So again, this is Venkatraman's PhD thesis, where he showed that it can fix nitrogen in pure culture. So they said that about 33% of the total soil algae in India are blue-green algae. Radhaprasana later said in one of our projects that it's almost 50%. So GSV and Nilakantan's paper in Journal and Applied Microbiology, again a very important paper. For the first time they showed that there's an interconvertible auxin inside a blue-green algae, and that vitamin B12 is also there, and it improves the growth of rice plants. But how does blue-green algae fix nitrogen? Because nitrogen is an anaerobic process. You all know that. Inside a root nodule, there is a nodule environment is anaerobic because you have leg hemoglobin, which removes the oxygen. In Azotobacter, you have a very high respiration rate so that it takes away the oxygen from the active site of the nitrogenase, or there is nitrogenase conformation so that the sites are protected from inside. Or you have azospirillum, which is microaerophilic. But how does blue-green algae fix nitrogen? So it happens in heterocysts, and inside the heterocysts, there is no oxygen because the photosystem 2, which is involved in oxygen, and this was done by BRC, by Joseph Thomas, another excellent scientist whom you all know. And then that solubilization of tricalcium phosphate by blue-green algae that they also do. And then there's an All India coordinate project on algae, and there's a lot of work on azolla at Cuttack, Coimbatore, Jorhat, many other places. And then the PL480 schemes, the first scheme on rhizobium was in 1969. They made a map of those rhizobium strains in India. There was an ASCRP on pulses, groundnut, and soybean. I think all of you know because you are all the centers of those things. And then the US soybean inoculants were inoculated in India in the late 1960s at Pantnagar and Jabalpur. And 12 serogroups, only 4 persisted after 3 years. What it shows is that the native rhizobia compete, the inoculated ones, and then only few will remain. There was a lot of work on radiation-induced mutants, and then a lot of work by Dr. Gaur on promiscuity of groundnut, which you know. There are 51 legume rhizobia nodulate. 51 legume rhizobia can nodulate groundnut. So it is so promiscuous, and then groundnut rhizobia in turn nodulates another 32 species. And then Dr. Kulkarni and others who are in groundnut, they have done a lot of work on similarity of these to develop strains. And lastly, I come to the genetical approaches. Because of the oil crisis, this was a bonanza for BNF. And there was an expectation at that time that the NIF gene transfer would result into nitrogen fixation in rice plants or wheat plants. And 31st of July 1975, if you remember the date, is a classic in the Nature, in a single issue of Nature, 3 papers were published. Gibson, Dilworth, and LaRue. That was the first time it was shown that rhizobium can fix nitrogen outside its host. So far the belief was that nitrogen can be fixed only inside a leguminous plant. So this was the first time it was shown that it can fix outside in a test tube by inducing arabinose and others, and anaerobic environment and things like that. And then at that time, there was a lot of hope that they will be able to generate nitrogen-fixing plants and cereals, genetic engineering for nitrogen fixation. And then in NRC in America, there was a soil, and there were studies by Dr. N.K. Notani at BRC, at Baroda by V.V. Modi, at IRA by H.K. Das and Sushil Kumar, and Hisar and Bengal and others. So I'm just now quoting those lines. What were the expectations? And the expectation is there till today also. This is very important to realize that nitrogen fixation is not a free process. It is not a free lunch. It involves a lot of energy. And actively, many families have actually done away with nitrogen fixation during the evolution of nitrogen fixation. So there's a time in scientific research when a number of developments coincide, making it possible to progress with a tough and complicated problem. A better understanding of photosynthesis, cell hybridization, plasmid, and gene transfer between cells, not necessarily genetic related, have opened new avenues of research. Although the news media often suggests that genetic engineers are on the verge of transferring nitrogen-fixing genes to non-legumes, this goal will probably not be achieved in the immediate future. So on and so forth. You can read this because when my paper is published, you can read from that. But what they never discussed was the yield penalty was never focused upon. You may succeed in transferring a nitrogen fixing capacity into a cereal plant, but then that will make it as a pulse. What is the difference between a pulse and a nitrogen fixing rice? I do not at least understand. Once a rice plant starts fixing nitrogen, then it becomes a pulse. Then there's no difference, and nature has already given us a pulse. So he says, although the news media often suggests that genetic engineers are on the verge of transferring nitrogen-fixing genes to non-legumes, this goal will probably not be achieved in the immediate future. 1982 he's writing. It's not done even today in 2025. Although the transfer of NIF genes into corn may be somewhat more difficult than yeast, the transfer itself should be feasible. Since it was a high-risk, long-range project, it should not be considered as a primary goal of developing countries at this time. However, it is important that work continue towards this goal. This is notwithstanding the fact that the difficulties involved were explicitly recognized in the just preceding sentence. Engineering the capabilities for the system to operate under low partial pressures of oxygen, to provide the energy needed for nitrogen fixation, and to integrate it generally into the system is a way that the genes will be expressed will likely be more difficult. Now I'll tell you my own personal story. This is all written. And I tell you there's nothing better than hearing. I think you'll all agree that there's nothing but. I'll tell you my own personal story. As far back as 1977, I wrote to Winston Brill. I was to join the University of Wisconsin at Madison for a PhD with him. I said, and he was working on transfer. I said, sir, this is going to involve a yield penalty. So the work that we are proposing to do, and he wrote back, probably it will take 10 years. I agree with you. It's not going to happen so soon. It's going to happen 10 years. And in all probability, the plant will be a very small plant, wheat plant. That's the same thing what you're telling, that even if you fix nitrogen in a wheat plant, it's going to be very small. It's not free lunch. And it's going to take 10 years. This is what Brill wrote to me in 1977. And we don't have even now today. So that was a goal which is pursued by pure researchers to understand the genetics of rhizobium, to understand the systems. But you have to get funds, so you have to convince the fund makers that big things are going to happen. And Postgate. I think Postgate you all know very well. Dixon and Postgate, the first people who transferred rhizobium nitrogen-fixing genes from E. coli from Klebsiella pneumoniae to E. coli. So I met him in 1996, and then even I mentioned that BNF is a very primitive trait. It has been lost multiple times. Plants themselves have done away with this primitive trait. They don't want it because it's the yield penalty. So anyway, that's another story. So what did Alexander say? Because I said now I come back to microbial ecology. Martin Alexander is a microbial ecologist. His book, all of you have read, Introduction to Soil Microbiology. He has put it very aptly. He says nitrogen fixation in agricultural land is rarely limited by a lack of highly active nitrogen fixing organisms. So where is the need to improve an organism? To make it excrete ammonia, or to have multiple copies of nitrogenase, or to have a high nitrate environment. And further, what does he write? It follows logically that a significant imbalance exists between the basic studies of nitrogen fixation by these microorganisms and the investigations needed to explain why so little nitrogen is fixed in nature. The enormous progress made in recent years in understanding the biochemistry and genetics of nitrogen-fixing organisms often leads only to improved organisms when no improvement is apparently needed. He is saying exactly the same thing which ecologists have been saying. He advocated the development of rhizobium strains able to survive the stresses on the seeds. So he said this is more important. Surviving the stresses is more important. He advocated the development of rhizobium strains able to survive the stresses on the seeds and soils, including predation by protozoa, and more competitive with resident rhizobia for nodulation, and also blue-green algae strains that are more competitive with other soil algae, herbicide resistant, less susceptible to predation, and as well as strains that are pest tolerant. And a lot of work showed, however, that the best of the native strains performed better in the field than the modified strains or exotic strains, showing the importance of the ecological adaptation of the strains and the difficulty of introducing an organism in an environment. And this echoes what Joshi had written in 1920. Attempts to grow better leguminous crops with commercial cultures of nitrogen-fixing organisms like nitrogen and nitrobacterin have not been successful in India. This shows that the organisms already present in Indian soils are efficient for the purpose and need not be replaced by those from outside. This is what we are all doing. So during post-oil crisis, as I said, there was an ASERP on BNF. You can read about that, in vitro activity, and HUP plus and HUP minus, which are all similar as inoculation of wheat and things. And international efforts in India on ICRISAT, you all know very well, N15 experiments with millets, pigeon pea nodulation, J.V.D. Kumar Rao's work on BNF, Rupella's work on high pea nodule selections, and then Lada's work on Azorhizobium epiphyticans. All this was done at IRRI, but then I thought I'll mention that. And then BNF work in stressed ecologies, what happens in under stressed environments, that the inherent effectiveness is more important than salt tolerance. These are some pictures of root hairs. I'll skip all these pictures because there's no time. These are some electron microscope pictures. And in the new millennium, we have diversified into vegetables and all this. I'll come to now the conclusions of my lecture because, done with the time. We have had culture collections, and then in my most recent paper, and this is for the attention of the Vice Chancellor of your university and the planners, because I thought this is the most important, because there's no use of my coming here and talking about the history. That is to tell people what is happening. What should we now do? Now, this is the most important. You should listen to this. This I have written in 2022. I think you all know Dr. R.S. Paroda, the DG, the former DG of the ICR. So for his 80th birthday, Dr. R.S. Paroda's 80th birthday, they brought out a special issue of the journal Indian Genetics and Plant Resources, the journal which NBPGR produces. They invited 80 articles from all over the world because he was an international figure, Dr. Paroda, you know everyone. And out of those 80, I was also one of them. So then I will write what is I want for microbiology, what is true to my heart. So I have said that there is neglect of rhizobial research and we need a blueprint for future efforts. And what are the blueprints? One is strengthen the existing ICR networks, crop and commodity AICRPs, and established 3 advanced centers of excellence. We need 3 advanced centers for excellence in our India. And who establishes them? I don't know. One, I called it as the India Nitrogen Fixation Research Center. Why? Because I established the India UK Nitrogen Fixation Center at UNSC with Oxford University. But that's only for 3 years, 4 years, we got funding and we finished. That's ended with that. So India Nitrogen Fixation Research Center. And another is an Indian Microbial Inoculants Technology Center. I have not talked about microbial inoculants. All the work, Dr. Brahmprakash and others, all the work on peat, lignite, liquid inoculants, all of you have done. This was not a talk on inoculants. This is talk on purely on biological nitrogen fixation. So I'm differentiating nitrogen fixation research, and I was telling Dr. C.R. Patil, head of the department, that in India, we do not have a single university, and I have begged from ICR for money and I never got, where we can have a good estimation of N15, not by isotope dilution, but by isotope abundance, because that's the only method by which you can properly measure nitrogen fixation. And you require a mass spectrometer, which is very, very expensive, but it can be done. So for this, you require money for the first one. For the second one, you don't require money. Indian Microbial Technology Center, this is already, you just need a name and you can establish that. And the last one is an India Soil Ecology Research Center. Because I've always believed, and during my whole talk, I think you would have seen the importance of soil ecology I was talking about. Soil ecology versus genetic engineering. So as I said, I made my efforts before I retired. It's for Dr. Krishna Raj and other youngsters who would now take it forward. So I'm coming to the last 2 slides of my talk. This is my talk. This is the end. So what is for the young boys and girls here? What are the new questions for the new century? How do we use the history of science in order to attempt to answer the new questions of the new century? As we ponder over new questions, we would do well to examine the implications of yesterday's science for tomorrow's. I'm reminded of Professor Martin Alexander of Cornell University, who wrote, kindly pay attention. Sadly, many researchers of the present and undoubtedly of the future, so he knew कि आगे भी यही होने वाला है. They're also not going to read. Sadly, many researchers of the present and undoubtedly of the future, as they consider upcoming issues and new directions, do not review what has been done in the past. Often such a perusal would reveal that the issues have already been proposed, good hypotheses have been tested and rejected, and worthwhile directions have been delineated on how to approach some of the topics. The error of not looking back while thinking ahead was aptly depicted in a statement heard at a seminar. This is Alexander writing. My research was confirmed by studies published 40 years ago. And this is my final thanks, besides the universities who have invited me, and my own regards to late professors in whom this memory has written. This is a personal one. I would like to express my everlasting gratitude to my own teacher and mentor, Professor G.S. Venkatramana of IRA, who at an early stage inculcated in me the immeasurable virtues of their generation. And what are those generations? Look back more respectfully. I think today's talk was only about that. Look back more respectfully. Look at the work done earlier. Look forward and look farther. Look deeper. Look more carefully. Look humbly. Look for beauty in our researches. My own reflections of over 5 decades in agricultural microbiology, being both a basic and applied science, have taught me the invaluable lesson of cultivating an interdisciplinary mind as an absolute necessity to appreciate the myriad phenomena at play in soil-plant microbe interactions. See, when you're talking about soil-plant microbe, there's no use of having an interdisciplinary team. I've seen in my institute, there's a soil scientist and a plant scientist and a microbiologist. Sir, we are 3 together working in this project. That is not interdisciplinary science. That is just you are doing your experiments. But in a single person, you have to inculcate the interdisciplinary bent of mind to be able to understand what is happening. Each one of them have to inculcate the interdisciplinary bent of mind. That's just an interdisciplinary experimentation. Interdisciplinary experimentation is to be distinguished from an interdisciplinary bent of mind. Thank you very much, ladies and gentlemen. I'm sorry I've taken so much of time. Oh my God. I've taken too much time.
sir, for that engaging lecture. Your words have beautifully sparked the curiosity and reflection in all of us. We will start looking for beauty in our researches as said by you, sir. Now it's our privilege to present a token of our respect and gratitude as a small tribute to his immense contribution to science and for being a part of our today's celebration. I kindly request all the dignitaries on the dais to felicitate our today's guest.
I'm extremely sorry that I forgot to acknowledge the presence of Dr. N.G. Patil, who's such a S.G. Patil, who's such an old friend of mine, and I'm really grateful that he came. The moment he heard that I had come, he came running, and I was taking the names of microbiologists, not of soil scientists. I'm so sorry, but thank you very much for coming. I also want to acknowledge, I did not mention adequately, although Dr. Alagawadi spoke about that we know each other and so many others. Dr. Bhagyaraj, I know for 40 years, such a long time. And, I'm so touched. When I informed him that I'm coming, he did not say anything. He just did not say anything. I did not know that he's coming. And then I was so surprised to know, I know him since 40 years from Hisar, from Lucknow, in England. So many meetings I've attended there in India, at Mau. And it's such a great pleasure to see you and wish you all the best for good health. Thank you.
We are deeply grateful to our dignitaries for honoring our chief guest and to Dr. D.L.N. Rao sir for accepting our humble tribute. This moment of felicitation stands as a reflection of respect, admiration and gratitude we hold for their valuable presence. We now come to a segment that recognizes the creativity and excellence of our young minds. Now it's my privilege to call Dr. Ashwini Ma'am, Associate Professor, Department of Microbiology, to brief about the events organized and announce the winners of respective events.
Distinguished dignitaries on the dais, scientists, colleagues, and my dear students. So as part of the memorial lecture series, we had some exciting competitions being organized. I'll quickly call the winners of these events. The first event was the cartooning competition. The theme given to the participants was microorganisms in everyday life. Okay, so I request the dignitaries to please honor these students. Okay, so the third prize winner is Anjuman Halyal, 4th BSc Agri student, AC Dharwad. Second prize is Sanjana, third year BSc Marketing, AC Dharwad. First prize goes to Neha, first year BSc Agri, AC Dharwad. Big round of applause. The next event was essay writing, and the theme was Microbes, the Hidden Creatures Shaping Our World. The third prize, again, Anjuman Halyal, 4th BSc Agri student, AC Dharwad. Second prize, Anjali, Senior MSc, Department of Crop Physiology, AC Dharwad. First prize, Kushan Kumar, Senior MSc, Department of Microbiology, AC Dharwad. Next event, we had organized the Rangoli competition. Third prize, Guna Maitri, second year, College of Community Science, US Dharwad. Theme was anything related to microbes. We have displayed everything outside. Please have a look. Second prize, Lakshmi Barchi, Junior MSc, Department of Microbiology. First prize goes to Sushmita, Junior MSc, Department of Plant Pathology. So another exciting event that we took was Agar Art. Okay, you can just have a look at the plates that we have put outside. Spirituality meeting science. Okay, so the third prize for this is backed by Chetan Kumar, Senior MSc, Department of Microbiology. Second prize, Baswan Gowda Patil, Senior MSc, Department of Microbiology. First prize, T.M. Monica, Senior MSc, Department of Microbiology. Congratulations to all the winners.
Our heartfelt thanks to Dr. Ashwini ma'am for efficiently and graciously calling out the winners and adding grace to this ceremony. I thank all the dignitaries for distributing the prizes. Having celebrated the exceptional talent of our students, let us engage with the larger vision that shapes the academic excellence. We have the privilege of hearing from none other than the Vice Chancellor of US Dharwad, Dr. P.L. Patil sir, to share his presidential remarks. Over to you, sir.
Good afternoon to everyone. So on the occasion of second memorial lecture in the series of our most respected stalwarts and institution builders, Dr. R.B. Patil, former Dean Postgraduate Studies in the university, and also Dr. T.K.R. Reddy, Head of the Department, Agriculture Microbiology, US Dharwad. On this occasion, the galaxy of dignitaries on the dais, respected Dr. D.L.N. Rao, the chief guest of today's program, respected Dr. Bhagyarajaji, former Head of the Department, Microbiology, GKVK, and our mentor, respected Dr. J.H. Kulkarni sir, honorable members of Board of Management, Sri Kundugolmat just left, and Sri Mantesh Shagotiji, organizers of the program, Dr. C.R. Patil and Dr. Krishnaraj, and all the faculty in the Department of Microbiology, all my senior teachers who have come over here, Dr. Alagwadi sir, all the students of Dr. R.B. Patil and also Dr. T.K.R. Reddy, who have come over here to witness this occasion, the former officers of the university, Dr. B.M. Khadi, Dr. S.G. Patil, Dr. Mantur, and all the faculty members, dear students. So this occasion is, I think, a memorable one for having organized jointly the memorial lecture in the name of two important microbiologists. So on this occasion, you have organized various competitions for the students. So I congratulate all the prize winners on this occasion. Hope this activity will continue. I think this is the first time that you have organized, isn't it? Last time was also there? Okay. So congratulations and compliments for continuously organizing, so that that's why so much of strength of the students still maintained. So they were curious in knowing their prizes and also their involvement. So unless we involve the students' activities along with this such a memorial lecture series or any functions, I don't think they are successful. So it is always better to involve the students in such activities by organizing such competitions, and themes are also very good. So microorganism centric. So I think we have to observe some of the rangolis or some whatever you have our students have performed. So on this occasion, after the inauguration and floral tributes to great scientists, the important event was the journey of both of them through a short video clipping, so well captured from the albums of both the scientists, and the reflections of Dr. Bhagyaraj, Dr. J.H. Kulkarni, Dr. Alagwadi, and also the other scientists with whom they have worked, both Dr. T.K.R. Reddy as well as Dr. R.B. Patil ji. So they share their experiences with them, and the most important one is, I think, the talking about history and presenting about history and teaching history is always boring. So I think today's the presentations on the history of biological nitrogen fixation, I think, so it is, so he created curiosity throughout his talk from the beginning. So early, late 19th century to up to 21st century. So he has covered exhaustively, and without, we don't know how, about 1 hour time. He was also surprised to know that how deeply he was involved in covering each and every aspect. Biological nitrogen fixation, the pre-independence era, and also he has linked to oil crisis about the biological nitrogen fixation. Nicely he has connected, and the post-independence, the research of biological nitrogen fixation, so which has been carried out in the erstwhile IRI, Imperial Agriculture Research Institute, Pusa, Bihar, and also further in IRI, ICR networks in in different institutions. Elaborately he has covered, I don't think he has left anything in the history of biological nitrogen fixation. He has nicely brought out the various aspects in that, and remembered all the scientists, both from Karnataka as well as in the entire country and also outside the country. So I think his elaborative digging of the history in the biological nitrogen fixation should be documented and must be published in a high-rated journal, I think. It should not go without a recording. So a lot of efforts he has put. So I learnt he prepares minimum of 3 to 4 months to give a talk like this. So give a big round of applause to Dr. D.L.N. Rao. So we never felt it is a boring talk. So such a talk I used to listen or got information that our late great pathologist Dr. R.K. Hegde used to do. History of plant pathology, so never the students get bored. So in the similar fashion, the history of biological nitrogen fixation, detailed in detail, he has given us. And at the end, he has given a task to all of us for establishing 3 Center of Excellences in microbiology. Hope our Director of Research, being microbiologist at the helm of the affair, he will make a team, at least one advanced center in microbial research, if not all. So let your effort come true along with all the faculty of microbiologists, and we have opportunity now from Government of Karnataka for establishing the Center of Excellence in various aspects. We have got the call letter for both the universities in Dharwad, Karnataka University as well as Agriculture University. It is an opportunity to put this idea, the Center of Excellence in Microbial Research, and more so the beneficial microorganisms. And we, as a human being, we should owe to microorganisms. Whether the history of microorganisms or biological nitrogen fixation is fixed into your brain or not, it is realistic that biological nitrogen is being continuously fixed in our soils, and it is blessing in disguise to all our poor farmers for nitrogen economy. So I think our center, university, starting from maybe the earlier stalwarts, I don't know. So I learned from Dr. J.H. Kulkarni sir, period as head of the department, and also Dr. Alagwadi sir, through whom I used some cultures. And I must tell sir here, I failed to acknowledge and he got annoyed. We should acknowledge from whom we take the material or from whom we take the help. The Department of Microbiology, since these following the footsteps of these heads of the department, further all the heads, Krishna Raj, then Jones, and now Dr. T.K.R. Patil, they are commercializing all the beneficial strains of biofertilizers, and they have also got license, and the production of biofertilizers is running like a factory in our Institute of Organic Farming. There is a lot of demand, so that's how we have commercially exploited the beneficial microorganisms in our institutions. Not only biological nitrogen fixation or biofertilizers, are also the organisms for the biopesticides. So I think the sky is the limit for making use of the beneficial microorganisms and commercialization, making their best use in the agriculture. So with these few words, once again, I thank all the dignitaries on the dais who have made it to attend this joint memorial lecture series. This will go jointly in future, and I request the organizers to kindly organize such memorial lectures in a befitting manner on specific days without fail. Thank you very much.
On behalf of all present, I extend my heartfelt gratitude to our Honorable Vice Chancellor Dr. P.L. Patil sir for his thought-provoking and inspiring presidential remarks. Your vision, wisdom and leadership continue to guide our university community and inspire generations of learners and researchers. As we draw curtains on this memorable gathering, to gracefully bring this luminous occasion to its conclusion, it is my privilege to invite Dr. C.R. Patil sir, Professor and Head, Department of Microbiology, to deliver the vote of thanks.
A very good afternoon to one and all. Respected dignitaries on the dais, respected teachers and my senior colleagues, student friends, families of our Ajits, families of our alumni association, and many of the alumni are there, my dear students. It's indeed a matter of privilege and an honor to be standing here to express our sense of gratitude and heartfully thank people who have helped us in organizing this event. I must admit that we were a bit lagging in organizing this. It was our Honorable Vice Chancellor, Dr. P.L. Patil sir, who kept on pushing us to, organize this. We have been trying, but we have evaded sometimes. And this time he did not allow us. He fixed up the date, he fixed up the speaker, and just told us to go with the organization. And sir, I really thank you for your constant support. Not only that, as you all know, he actually brought this under the university function, and now we don't need to collect money for organizing this. University itself has made arrangements for collection. So thanks to our Honorable Vice Chancellor for his support, the vision for honoring our institution builders. I also take this opportunity to thank Dr. D.L.N. Rao for such an engrossing talk, for having taken the pain to travel all along and share his thoughts with us. On behalf of the university, on behalf of the department, and on all our behalf, sir, thank you very much for taking your time and being here with us for delivering this memorial lecture. I express my sincere thanks to my teacher and perhaps a very senior microbiologist amongst all of us here, Dr. D.J. Bhagyaraj, who despite, the age doesn't matter, they all motivate us. Sir was kind enough to consider our request and, be a part of this function today. And sir, thank you very much for coming all the way and being a part. We missed you last year, but thanks for coming this year. I must also thank our, we always call the senior most microbiologist, Dr. J.H. Kulkarni, former Vice Chancellor, who has been a guiding force and who has always, constantly helped us in organizing this. In fact, it was his efforts to call individual seniors and, make sure that they participate. Sir, I appreciate, thank you for your support and thanks for participating in this event. Ladies and gentlemen, our members on the Board of Management have constantly been encouraging and supporting us. Today we have two of them here with us today. Sri Maltesh Shagoti sir, who is kind enough and very friendly man who came here to grace the function. Sir, thank you very much. And another board member, Sri Basavaraj Kundugalmat, who just left us for another function which was scheduled earlier. Thanks to both of them. I express my sense of deep gratitude and thanks to our Dean Agri, Dr. Kuligod sir, for providing all the facilities and helping us to organize this function. I also take this opportunity to thank Dr. Krishnaraj for all his support and help. Um, I will fail in my duty if I don't recognize the constant support I get, and most of us at the department are guided by our elders, including Dr. Alagwadi, Dr. Jagdish, Dr. Savalagi, and many others who were a part of this. Wonderful alumni of the department who have been very, very instrumental in contributing to organize these functions. Just one instance of saying how we get connected, the reminiscence, that 2 minutes video was actually a connection of our alumni, our colleagues at the department and the present students. So we got connected to an alumni in Tirupati who went to Dr. T.K.R. Reddy's house, dug out all the old photos from his house and sent us, and then my colleagues organized it. My students at the department and collated it and made a beautiful video. This was done in a couple of days. So it just speaks about the department connect, and I'm thankful to all of them, the alumni, the senior colleagues. Sir, my students of the department and my colleagues at the department have been toiling hard for a couple of days now. I express my deep sense of gratitude and thanks to each one of them for their support, standing at all times to organize this event. Thanks to each one of them. I understand many seniors, former officers of the universities, my teachers from US Bangalore, many have taken interest to be a part of this event. Thanks to each one of you, and I appreciate if you can continue supporting us like this. We'll be able to organize more and more of these memorial lectures. Thanks to the staff who have been helping me in cleaning this up and organizing this. I sincerely acknowledge my supporting staff at the department, secretary staff at PPMC and Vice Chancellor's office who have been also helping me in organizing and helping my team in bringing up this show. Thanks to each one of you. If I have missed anyone, it is not an intention. It may be a mistake. Please forgive me for that. Thank you one and all for gracing this function, and I once again look forward for your support in organizing in future too. Thank you very much.
Thank you, sir, for your eloquent and graceful words of thanks. You have woven together the threads of today's celebration. Before closing, it's our duty to honor the spirit of our nation as we play the national anthem. I request everyone to stand together for national anthem. As today's achievements settle into lasting memories, let us frame this collective moment. I kindly request all dignitaries, faculties, alumni, and students of Department of Microbiology for a group photograph. With smiles framed and memories scaled, we conclude this beautiful gathering.